Thank you, Judith. Judy, for a great talk. All right. Okay, for the last talk of the day, I'd like to introduce Professor Thomas Nutman, who's the chief of the Laboratory of Parasitic Diseases at the National Institute of Allergy and Infectious Diseases. So he's really a leader in the field, and he works on the host immunology of parasitic infections, which is a totally different angle. So I think it will nicely complement the session. Looking forward to your talk.
Thank you very much. And thank you very much to the organizers for inviting me today. And it's also a pleasure to share the program with two people I've known since each of us, I think, was a trainee. So have a long history. Although our lab works on a number of different aspects, today I'm going to talk about the host response to human filarial infections, but also the consequences of these infections for responses to other both diseases and call it non-parasitic vaccines. And I think we can learn a lot about both parasitism and vaccine development by having a detailed look at the host response and really understanding what actually happens. I should say that I come from a clinical background initially, and I think the other piece is that we can never really lose sight of the patient and the human cost, as was so eloquently discussed by Peter. I'm going to skip this because we've talked about that. And most of my research career has been on studying these filarial infections. Does this, if this works? But suffice it to say that there are 8 filarial infections that commonly infect humans, and the top 4 there, 3 of which, or all 4 of which were mentioned by each of the speakers, which were Bancrofti, Brugia malea, the 2 causative agents of lymphatic filariasis, Onchocerca volvulus, the causative agent of onchocerciasis, and Loa loa, the African eyeworm. And we certainly know that these are collectively the second leading cause of disability worldwide. These are neglected except by German metal bands and in the bottom, over there by the Shin's latest album, which says heartworm. It's the dog Dirofilaria. This was already covered by Judy, and I will not dwell on this at all. All of the filaria have a similar life cycle. The difference is largely in where the microfilaria, which are these little the L1 stage that are found in the blood or the skin, in terms of their location and the vector that transmits it. And basically, the life cycle is largely the same. Infective stage larvae are deposited in the skin. They develop over 6 months or so into adults. They mate and produce microfilaria. And it's these microfilaria, largely, that are responsible for the pathology associated with this infection. So with filarial infections, there are some concepts that really are make the study of these really relatively intractable. There is no permissive small animal host for any of these parasites. So that's, with the exception perhaps of Brugia malei, which can be found in naturally or spermally in gerbils, but none of them go into and complete their life cycle. None of them induce the kind of pathology that you can see, so it becomes really difficult. The study of immune responses in filarial infections then requires population-based field studies or very detailed analyses of patients that might get referred to people like me in major medical centers. And so anything that we want to do in terms of parasite material, the genomics of these parasites, which we have solved, fortunately, all of the parasite material generally must be obtained from human tissue. So this is a mess. But our approach to the study of these infections has really been broad-based, and we've sort of focused on the host-parasite interaction, and we really look at a whole number of different things, from the clinical to the basic to the field-based, and we know that these are all completely interrelated. And what I'm going to talk about today is really work on our host side of things. We can use this, to study the human host in terms of immune regulation, natural history, pathogenesis, diagnosis, treatment, surveillance. But what I'm really going to talk about today is sort of the immune response and how it's regulated in these infections. Now, through many sort of years of studying patients, when you think about the parasite-host interaction, what we found was that 80 to 90% of people who were infected worldwide end up having a subclinical patent infection, meaning they have relatively high levels of parasites in their blood or their skin. They have no obvious clinical manifestations of this infection, and as I'll show you, this is a reflection largely of having an appropriately modulated immune response or an inflammatory response to the parasite. And if you think about it, this is really the perfect host-parasite interface, right? The parasite doesn't cause huge pathology in the host. Because of that, it survives, and it allows it to be transmitted, in this case, the microfilarial stage, to its vector. And as I've been thinking about this more and giving these kinds of talks more and more, what I find is that this is actually not just filarial related. This is probably true of the intestinal worms. It's actually true of things like malaria, visceral leishmaniasis, where the overwhelmingly large burden of infection is clinically silent. However, what gets all the notoriety, and appropriately, is the pathological consequences of these infections. And these, we think, occur because there is no brake on the immune response, or the uncontrolled response, the pro-inflammatory response, and all the pathology that, as far as we can tell, that's associated with this infection is immune-mediated. So in the case of lymphatic filariasis, the elephantiasis, or the massive scrotal edema here, or in the case of onchocerciasis with the sclerosing keratitis and blindness, and the severe deforming skin infections, shown here is the leopard skin, or in loa loa, which is these calabar swellings, or a picture of a heart from a patient who has endomyocardial fibrosis associated with the massive eosinophilia associated with loa loa. So this is our paradigm, and I'm going to try to show you sort of what went into sort of understanding this, what I hope regulates this, and what the consequence of that regulation is. So this summarizes maybe the first 10 years of my life in one table. And this is actually the data that sort of define that whole idea of what a modulated immune response that drives the subclinical form of these infections. So if you look at, in this case, T-cell responses to parasite antigen in these subclinically or asymptomatic infections, what you find is they fail to produce, to proliferate to parasite antigen. They fail to produce IL-2 or interferon gamma. They make moderate amounts of IL-4 and IL-5, and their responses to non-parasite antigens are largely intact. So this is not an immune defect, a global immune defect. This is a parasite-specific downregulation or inability to mount a response of a particular nature. If you have pathology associated with this infection, and it doesn't matter which of those infections you look at that I mentioned, you find that all of these mount this massive pro-inflammatory, in this case, T-cell response, without any compromise to their non-parasite response. And then if you use, and I would sort of not warn, but mention that if you look at these patients who were, and we coined the term putatively immune almost 32 years ago, I think, if you use these patients who have lived in endemic areas, have resisted infection for their entire life, and you look at their response to parasite antigen, a T-cell response, they mount a really quite dramatic, polarized, now we call it a type 1 response, to the parasite. Again, with pretty good response to non-parasite antigens. And I would say that when you think about those beautiful pictures that Judy showed with the very big worms, and you think about things like even TB or HIV or malaria now, we really understand that it's actually not antibodies that's driving a lot of the protection, but it's actually T cells. And so when one thinks about vaccine development, one also needs to begin to think about the fact that the T-cell arm needs to be thought about, and it just can't. It's not going to be like a virus where you get a neutralizing antibody and you protect. So anyway, just a side note. So that was the first 10 years of my life, and the next 10 probably can be largely summarized here. And now what I wanted to talk about is that this puts in the context of actually what happens from the beginning of infection, early infection, to the decades-long infection that you have. And some of this is inferred from animal models because we never know when humans are actually infected. But suffice it to say that in the first early piece of that, as the L3s are developing into adults, that you actually get a robust response, a CD4 positive T cell response to parasite antigen, shown here in red. And then at the time of patency, that is when the microfilaria start being produced, this T cell response is downregulated. And it remains downregulated until you actually treat individuals with antifilarial chemotherapy. The response to non-parasite antigens, we'll call them bystander antigens or allergens or vaccines, is largely intact over most of the infection. But over time, you end up having a spillover of antigen-specific downregulation that occurs. And what we found very early on is that when IL-10 was discovered and we had antibodies to it, that you could reverse some of that with IL-10. And this is going to come back. I'm going to come back to that in a second. So the third 10 years of my career was to sort of fill in all of these things. And really what we now know is that in the first 2 weeks of infection, because these are skin penetrating, that there's a host of cells that are largely innate cells, ILCs, eosinophils, macrophages, dendritic cells, all of which are in the skin and the draining lymph nodes of the skin, are activated, and we now know what are some of those signals that drive those. But what they ultimately do is they direct the immune response to the parasite. And at the time that these worms are developing at sort of in this up to somewhere between 2 weeks and 6 months, there's sort of a balanced immune response to the parasite, Tregs, Th1s, Th17s, Th2s, with all their appropriate cytokines. There's antibody response. But when the parasites produce these microfilaria, and these are really the driver, the major antigenic burden of these infections, something happens to the immune system such that the Th1 response contracts completely, very dramatically, as does the Th17 response. There's an expansion of Th2 responses with IL-4, IL-5, IL-13, IL-10. The IL-4 drives the development of these M2 macrophages, alternatively activated macrophages. And there's a marked expansion of these T regulatory cells, and making largely IL-10 and TGF-beta, and some other things. Over the long term, even these Th2 cells or populations contract. These T regulatory cells take over. And as a consequence, the T central memory and effector memory cells, these cells that have to be poised to respond to new infections, are also markedly downregulated with sort of a profound, not a profound, but a markedly diminished function to respond to parasite antigen. And this has consequences to the response to these bystander antigens as well. So sort of some experimental data to sort of go along with my cartoons comes from the studies, again, of patients. And shown here is just a heat map of what turned out to be a microarray experiment where we had the ability to study travelers who had relatively acute infections versus patients who were born and bred in the endemic regions. And we purified CD4 positive T cells in this case and did microarray analysis. And the bottom line here is that with chronicity, long-standing infection, what happens is that you get this markedly downregulated pro-inflammatory response. This is one of the pathways. This is the NF-kappa B pathway. But the bottom line is that this chronicity is what, and I'll call it chronic antigenic stimulation, is what we think is driving this sort of downregulated immune response. Now, some of these studies are really not easy to do, and I think this is one of the things that sort of nobody who does what we do completely understands how difficult doing, let's call it, first world immunology elsewhere. So to do some of these immune studies on patients, we end up having to go to the end, largely to the end of the road. And this is in the dry season, so it's actually relatively easy to transport. We have to set up its lab without electricity in order to process the blood, to prepare things for things like flow cytometry and some other things that we can do. But when you have these kind of situations, you have to come up with sort of techniques to kind of do things kind of in the field without refrigeration, without a lot of technology. But you can overcome these relatively easily, we think, these days. And with that, we were able then to study these patients and really begin to pick apart the nature of all the cells that are being driven by this infection. Shown in the red are patients with the filarial infections, and then neighbors of theirs who were age-matched without the infection. And we were able to do multicolor, 12, 15 color flow cytometry of all their cell populations. And I just focus here on the fact that what happens in these filarial infections is largely you get this massive expansion of CD4 positive cells that just produce IL-10. And these are what are called the adaptive Tregs. And it's this IL-10 producing population largely, we think, that is responsible for all that downregulation that actually occurs in this infection. Now, the other thing that this does is that it sort of sets up this sort of a way to understand how this all gets set up. And I just and what we and many other people in the field have sort of figured out is that there are different parts of the puzzle that are being provided by different pieces. So the worms themselves, and Peter mentioned some of this already, we know secrete. They secrete TGF-beta homologs. They secrete, in this case, an HSP molecule that actually binds the human IL-10 receptor. They produce cytokine-like molecules themselves. They produce a number of secreted molecules that have profound immunological effects. The host, in response to that, makes largely these alarmins, IL-10 again, downregulated inhibitory responses. They modulate TLR expression on antigen-presenting cells, and you end up with this alteration of antigen-presenting cell function, which ultimately leads to an anti-inflammatory bystander suppression. You drive Treg responses. And this is sort of the general sort of state of affairs. Ultimately, if you fail to sort of modulate this parasite-specific response, you end up with pathology. And we're trying to sort of figure out where all these things fit in. So I want to return to, so that's sort of the state of sort of the host response to the parasite. And I want to move very quickly to really the consequences of this. And the consequence of this is what I have here in the blue, this response to bystander antigens or allergens or vaccines or other pathogens. And I'm going to give you just a couple examples of this as well. So if you read the literature, at least the current literature, and there's a dogma, and that is that if you have a Helminth infection and you have something else, that the Helminth infection has the ability to modulate, you name it. Right? We know that there are data that it modulates atopy or allergy or asthma. I'm going to talk a little bit about the responses to vaccination. In general, people are talking now about how it really makes a difference in terms of the outcomes of non-Helminth infectious diseases, including TB and malaria and HIV. And you can imagine why all of us who study these worm infections have moved to studying these co-infections, because that's where, everybody's interested, right? The inflammatory autoimmune disease, HIV, malaria, there's money for that. And so if it's a hook in a sense. This is a study we did many years ago, and it's been replicated multiple times. And this is a study that we did in an onchocerciasis endemic area, and we've repeated it in the lymphatic endemic area. And this was taking patients with onchocerciasis, those are the infected patients, and those without onchocerciasis living in the same area. And we vaccinated them to a recall antigen, in this case tetanus toxoid. The tetanus vaccine that we all used to get, and we looked at the patients at the pre-vaccination and then 6 months later. The response to PBD is just a control antigen in this population, and basically at 0 and 6 months they look identical. In this case, when we looked at titers of anti-tetanus antibodies, they were largely the same in both groups. But if you look at the response, this is the T-cell response here to tetanus in the pre-vaccinated individuals who didn't have onchocerciasis and those who did, you can see that at 6 months after vaccination, their T-cells, in this case, proliferated markedly to the tetanus toxoid. And if you look here over at the infected individuals, they basically barely responded. And this is despite the fact that they induced at least a neutralizing antibody, enough of an antibody response that would consider a vaccine to be a take. Moreover, if you actually now looked at the cytokine profile of these, what we found was that if you were filarial infected in this case, the response to tetanus toxoid was largely dominated by a type 2, didn't matter which one we measured, but here we're measuring IL-5 and IL-10, which is a markedly downregulating cytokine. And their response to their interferon gamma response, which is the sort of hallmark Th1 type response, was essentially absent. And that is the complete opposite of what happens if you were filarial uninfected. And they didn't have other worm infections in this case. And so what this tells us is that it's the quality of the immune response that's being shaped to a bystander antigen. And we've repeated this with oral cholera vaccine and hepatitis B in vaccine. And it's pretty much a very sort of profound kind of and constant sort of finding. So typically you get a response that's measurable, that's appropriate in terms of antibody response, but the nature and it turns out the duration of the response is markedly diminished if you have a pre-existing Helminth. And if I were not just making, if we were not just thinking about doing parasitic infections, but if you're thinking about doing TB vaccines or new vaccines in disease Helminth endemic countries, I would absolutely treat the cohorts with anti-helminthic therapy prior to putting them under vaccine trials. And this is in fact being done in certain regions of the world as we speak. In the last few minutes, I'm just going to talk about one other area that we think that is very important, and that's TB. And it was raised earlier this morning, and I won't talk about the importance of TB as a global problem, but I just want to point out for those of you who don't know that there really are two forms of the infection: the dominant form, which is latent TB, and the active form, which is responsible for all the deaths. And what you probably assume, but this shows in a picture that the helminth infections and tuberculosis have a broad geographic overlap, and you could put HIV here, you could put malaria here, it doesn't really matter, but the bottom line is that for TB, it's certainly the case. And moreover, as many of you probably know, BCG vaccination is used worldwide, and particularly in Northern Europe. But in all of these areas that are helminth infected endemic, vaccination has the poorest protection rates. And so there's some sense that there's an interface, not causality, but let's just say that we have to keep this in mind. And so we actually have done a lot of studies to sort of look at this and look at the interactions of these two infections. And the other thing from an epidemiological point of view is that people actually get infected early in life, that is with the parasites, and they get infected with TB typically later in life, or certainly later than early childhood. And so we've been working on what is the interface between these two types of infections, and we think that a lot of this can be understood by understanding the host response to each of these. And the beauty, of course, is that we have antigens, both TB-specific antigens and helminth-specific antigens, to begin to pick apart this in a little bit more detail. Broadly speaking, it's not surprising if you look at the chronic helminth infections that's dominated by IL-10 and TGF-beta and some of these type 2 cytokines. And then you look at TB, which is largely dominated by type 1 and type 17 type cytokines. You can see that there might be an immunological interaction that might have an impact on the control of TB. Whoops, sorry. And this is actually demonstrated, with data. So here is mycobacteria-specific responses in filarial infected and uninfected individuals. Filarial infected in green, filarial negative and uninfected in red. And now you're looking at cytokine production in response to PPD or a response to whole MTB organisms. And you can see that the type 1 responses, the interferon gamma and the IL-17 that is absolutely critical for control of TB and maintenance of the latency stage is markedly diminished in these filarial infected individuals. And moreover, these multifunctional T cells that are these double and triple producers, so the interferon gamma IL-2 or interferon gamma TNF or the triple producers interferon gamma TNF and IL-2 are similarly markedly downregulated in filarial infected and in this case strongyloides, which is a soil-transmitted helminth infection, compared to those people who are uninfected, all of whom have latent TB in this case. I'll skip that in the interest of time and say that now we have the ability to sort of look at this in very broad strokes using multi-parameter flow cytometry. Suffice it to say that if you look at uninfected or filarial infected individuals and you look here at their infector memory cells or their central memory cell population in response to the parasite antigen, if you don't have filarial infection, you largely have a response that's dominated by TNF. In the case of the parasites, it's largely dominated by IL-4. The response to CFP10, which is a mycobacteria-specific antigen, It looks quite similar in terms of if you have a filarial infection, the quality of your memory response is really different compared to those who don't have an infection. And it's not global because the response largely to mitogens and superantigens are identical. The other thing that I'm just going to say is that IL-4, which is driven by the parasite, drives alternatively activated macrophages. And if you as you probably know, macrophages are the host the major cell that mycobacteria infects. And so with filarial co-infection, you're driving the induction of these alternatively activated macrophages. If you actually infect alternatively activated macrophages versus these classically activated or M1 macrophages with mycobacteria, in this case BCG, you can see that using amnis in this case, that the alternatively activated macrophages internalize the mycobacteria, whereas the classically activated macrophages do it less well, and this is just a quantitative look at that. It turns out that these alternatively activated or M2 macrophages handle MTB and in this case BCG very differently. If you look following infection, you can see that the concentration of IL-10, which again is bad for MTB because it reduces the interferon gamma necessary to control it, is markedly elevated at about a log-fold more in these alternatively activated macrophages. And this just points out how IL-10 is detrimental to TB progression. IL-10 in macrophages signals through two pathways, a TLR-dependent pathway and a TPL2-dependent pathway. And so it turns out that if you look at TPL2 upregulation, it turns out that if you look at the TLR pathway, there's no real difference between the two types of cells. But here, TPL2 is driven by the mycobacteria very early, and at the same time that the TPL2 is driven, there's a marked induction of IL-10, and that's not the case for the classically activated or M1 macrophages. You can do this by Western blot as well. And you can actually inhibit TPL2 in these alternatively activated macrophages chemically, and you can now to essentially inhibit by 100% the IL-10 production. So we think we've actually used this sort of model of co-infection to really begin to understand how some of these processes that we're seeing actually get set up. So finally, and I really have done very few mice experiments in my entire life, we actually set up a model to actually mimic this co-infection of where we make mice microfilariemic. We infect them with virulent TB by aerosol, and then we followed them over time. I'm just going to make the point that in the lungs of these co-infected mice, there's a marked expansion of IL-10, and when you take the lung tissue, and in this case we did a microarray, again TPL2 was markedly elevated. And so we sort of went from the clinical observation to sort of understanding how this IL-10 is actually working in the context of a true non-parasitic infection. Okay. Very lastly, we then took this to the field and we said, okay, if these co-infections really make Helminth infections really make a difference, can we look at the progression of latent TB versus active TB? And this was a heroic study by a number of people, including my large number of colleagues in India, where we followed 5,000 people in four very large communities over a 7.5-year period, and every 2 years we essentially evaluated them and classified how many people went from latent TB to active TB. as a function of whether you had a Helminth infection at the beginning of the study. To cut a very long story short, despite the fact that all this immunological stuff was happening on the face of these worm infections, in the end, the incident TB between the infected and uninfected individuals was largely the same. And so we're left with this sort of conundrum of, having these immunologically relevant sorts of findings with a relatively minor effect on outcome in terms of disease. So the jury is still out. So the take-home messages of what I'd like to leave you with is that chronic tissue invasive Helminth infections, and these are not just the filarial infections, induce a parasite-specific modulation of the immune response by a number of mechanisms that serve to limit immune-mediated pathology. And central to this modulation is IL-10 production, and I should say that we now know that some of the other IL-10 family members, superfamily members, are also responsible for this. These chronic tissue invasive Helminth infections do drive immunologically measurable bystander effects on vaccine responses and responses to other pathogens, but clinical outcomes, and particularly in those infections and conditions dominated by significant inflammation, such as active TB or an acute malarial attack, remain largely unaffected by these coincident Helminth infections. And I think with that, I'm going to stop. The stuff I talked about today was really done by a number of people over many years. The four that I'd like to mention are listed there: Subhash Babu, Kathy Steele, Roshan Exonani, and Soumya Shoms Chatterjee. We had help from our group in India, and the people are listed there, as well as some of the work that we did in Mali with our colleagues there. And I think, like everybody else, it takes actually more than a village. It really takes lots and lots of people. This is my current lab, our group in India, a collection of people that work with us in Mali. And really, we cannot emphasize enough the sort of absolute kind of heroes in all of this work, and that's the patients who've been willing to, and subjects that have been willing to sort of provide material and hopefully some insights into what we do. So thank you.